Hi guys, in this video we are going to see about the history of immunology. We all know that we have soldiers inside our body, that is the immune cells. So whenever an antigen enters our body, they just go and kills the cells in their own style, because some secretes toxin and some just go and engulf the antigens. And we know that Louis Pasteur is the father of immunology, and we know few scientists' names, but we don't know much about the history of immunology. So let's see who made the history. The history started from the great Thucydides in Athens 430 BC, who pointed out that the people who were once attacked by the plague could not get the fever of the second attack of the disease. Based on this understanding that immunity is produced after the attack of a disease, the crude practice of inoculation began in the beginning of 15th century. Inoculation is nothing but the process of introducing microorganisms into an organism to produce immunity. In this method, the smallpox lesions were scrapped from the person suffering from the disease, and the material cord was introduced into a healthy individual with the help of needles. This method is highly risky because some survived and many died due to this disease. Later, this process was known as variolation, as smallpox was known as variola at that time. Next comes the Edward Jenner, an English physician who had suffered the pain of variolation at his younger age. He observed the milkmaids who often contracted cowpox were remarkably resistant to smallpox. So, in 1786, Jenner inoculated a boy who volunteered himself for this experiment with material got from the lesions of a milkmaid who was attacked by cowpox. After some weeks, the boy was again inoculated with infectious pus taken from a patient suffering from smallpox, but the boy did not get the disease. This made him to conclude that this process of inoculation leads to immunity against smallpox. Next comes the father of immunology, a French chemist, that is the Louis Pasteur, who coined the term vaccine for such process of prophylaxis, the prevention of disease, in honor of Edward Jenner, who prepared the first vaccine from cowpox. Louis Pasteur worked on the common diseases such as pebrine, anthrax, chicken cholera, and rabies. He inoculated chickens with an old culture of bacillus, and the chickens failed to develop illness. And this aged culture became the first attenuated vaccine. Then Pasteur cultivated anthrax bacteria at a higher temperature of 42 degrees Celsius and inoculated to a sheep, and the sheep did not develop the disease, because the pathogenic bacteria lost their virulence on cultivation at high temperature. The most interesting of Pasteur's work was the production of vaccine for rabies. This method is based on unnatural host. He injected the spinal cord extract from rabid dogs into normal healthy rabbits. In these rabbits, the spinal cord is infected with rabies. Extract was prepared from the infected spinal cords and dried for several days at room temperature. This extract contained the attenuated viruses. The rabies vaccine was prepared from this. Mechnikov is the Russian zoologist, discovered the cell-mediated immunity and phagocytosis. While observing the transparent starfish larva, he could see some motile cells which he thought might be protective in a digestive way, and he called these cells the phagocytes. Later, he observed the phagocytic cells in Daphnia that ingest and digest yeast that are pathogenic to the Daphnia. In 1908, Mechnikov shared a Nobel Prize with Ehrlich for his contributions to immunity. Ehrlich, a German scientist, found out that immunity could be transferred from mother to the offspring, the side chain hypothesis, discovered the mast cells, developed the concept of horror autotoxicus, the mechanism whereby the body does not react immunologically to its own cells and tissues. Also renamed Jules Bourdin's Alexin as complement. Now, who is this Jules Bourdin? So he is a bacteriologist. He showed that the human blood normally contains a group of heat-liable substances which are strongly bactericidal when antibody is present, and he named it as alexin. He also showed that these complement is absorbed and made inactive or fixed by the antigen-antibody complexes. This complement fixation becomes the basis for the laboratory identification for many disease-causing organisms. He won the Nobel Prize for his contributions in 1990. He also studied the formation of anaphylactic poisons produced at the time of complement fixation. While talking about the anaphylactic poisons, before him in 1913, Charles Richet, a French physiologist, was awarded the Nobel Prize for his work on anaphylaxis. It is nothing but the hypersensitive reactions of the body to foreign proteins. Next, in 1818, George Nuttall, an American bacteriologist, found out that defibrinated blood had bactericidal effect, showing the presence of serum substance in blood. Following him, in 1894, Pfeiffer proved this through his experiment Pfeiffer phenomenon. The greatest support for humoral theory came from the studies of Emil von Behring, a German bacteriologist. For his work on antitoxin and the principle of antiserum therapy, he was given Nobel Prize in 1901. Next comes the Ambroth Wright. He and Stewart Douglas formulated the theory of opsonization. Wright termed the serum substance as opsonins. It is nothing but an antibody that attaches to a particular antigen and prepares it for phagocytosis. Next comes the Robert Koch, a German physician and pathologist, was awarded the Nobel Prize in 1905 for his research in tuberculosis. And we are familiar with Alexander Fleming, so he is a British bacteriologist, received Nobel Prize in 1945 for his discovery of penicillin. Next, it is Vidal Fernando. So he devised the blood test known as Vidal's test for the diagnosis of typhoid fever. In 1959, Bernard Macfarlane formulated the clonal selection theory of antibody formation, and he shared the Nobel Prize with Peter Madaber in 1960 for the discovery of acquired immunological tolerance. In 1957, Isaac and Lindemann discovered the interferons. Next, it is the well-known Austrian scientist Karl Landsteiner. So he was awarded the Nobel Prize in 1901 for his discovery of blood groups. He coined the term hapten in 1921 to refer small chemical groups that become immunogenic only when added to a carrier molecule. In 1943, with Merrill Chase, he discovered that a class of allergy known as the delayed hypersensitivity could be transferred from the allergic animal to a normal recipient with lymphocytes. In 1972, Rodney R. Porter and Gerald M. Edelman received Nobel Prize for their fundamental studies in the chemistry of immunoglobulins. In 2011, Ralph M. Steinman received Nobel Prize for the discovery of role of dendritic cells in human immune response. Again, in 2011, Beutler and Hoffman received Nobel Prize for their work on toll-like receptors. Few major scientists' names and their discoveries are mentioned here. The history doesn't stop here. Still, researchers are going in this field. Thank you guys. See you in my next video.